So, this we have seen. So, we will get back to von Bayer's principle. So, we are in this comparative embryology and then transitioning into evolutionary embryology pretty much in the same topic. So, just this I am showing just so that you remember the continuity. So, we went through all of this. So, you will remember this preformation, okay. The whole structure is in the sperm head, was in the sperm head, no longer, just to be abundantly clear with the facts. We went through this and von Bayer's principle. I felt that I went little faster through the von Bayer's principle last time. So, this time we will go more carefully, and I want to make sure you understand it, because this is a really a crucial set of concepts that bridges developmental biology and evolutionary biology. So, therefore, we need to really get this clear in our mind. So, the primary point, all four to put together, the primary point is, if you are going to say a more developed organism comparatively than another one, it does not mean during the embryonic development of this more complex organism, you are going through the adult stages of the less complex organism that existed much earlier. Okay. Like for example, today's birds and reptiles do not go through the adult stages of dinosaurs, obviously, right. Okay. So, that is the main point. For right now, having learned whole lot of molecular biology, evolutionary theory, etc., this looks obvious, but it was not actually. At the time von Baer proposed this, there was an earlier competing theory which was exactly saying the opposite, like the embryos go through the adult like structures of less evolved organisms. Okay. So, that is the theory that was there for long, and quite ironically, while this theory these principles supported Darwin's theory of evolution, von Baer spent his entire life opposing Darwin's theory of evolution. While Darwin got very excited how this helps him to find support, which we will see in detail, because that I really want to emphasize, because it is an important point. So, this kind of pictorially tells you the same thing that we saw in the previous slide. Okay. So, if you look at human, marsupial, chicken, salamander, fish, etc., if you look at the embryos, very early embryos, they look more or less similar. If you take closely related two different species of a given genus, if someone has not labeled the bottles in which the specimens are, you will not be able to distinguish them, very early ones. But as they go through, the species specific specialized structures become clearer, as the development proceeds. So, this was very exciting to Darwin, because this gives you two important points. One is that the very early embryo resembling very early embryos of diverse organisms having similarity, in essence the embryonic similarity supports common origin, descent from a common ancestor. And as the development proceeds, specialized structures start they evolve and they become clearer as the development progresses. That supports adaptation, species specific adaptation. So, actually you are seeing two interpretations both consistent with Darwin's theory of natural selection driven evolution. So, your starting material is the same, but then you are modifying its development. So, alterations in development is the point that gives the required variations for natural selection to select. So, that is what this supports, and that is why Darwin was very excited about. So, and another point is that evolutionary relationships become more obvious when you look at embryonic or larval stage rather than looking at the adult stage, and that is very well illustrated in the two organisms shown here. If you look at the first panel, barnacle and shrimp, both of them are crustacean arthropods. Okay, these are jointed legs living in the ocean. Okay, that much zoology, if you know, that is good enough for now. And larval stage, they look significantly similar. You know, one might look little smaller and less complex, but overall they are very similar compared to the adult. So early taxonomists classified this as a mollusk, okay, because its structure, outer structure resembles that of mollusks, and this one looks very different. But while shrimp and barnacle at larval stage, they reveal their common ancestry. So this is what Darwin pointed out. So we need to look at the embryonic stage for accurate classification of organisms based on their evolutionary relationship. So the modern taxonomy groups organisms based on that. Okay, the evolutionary relationship is shown in the way we classify organisms. So that is one of the main outcomes of von Bayer's theory when you look it from the evolutionary perspective. So we are going to stay with this theme with the 2 or 3 more examples. So here is one. So this is an embryo of a salamander. Don't worry about what salamander is. It's just the surface is opened and the pharyngeal arches, basically this part. Okay, so is shown highlighted in this purple color. So group of cells that form these structures in the early embryo give rise to these gill arches and this hyomandibular in the fishes. These 2 bones support the 2 jaws. Okay. So they themselves are not supported without this hyomandibular structure in fishes, and the same embryonic structures or the cells, pharyngeal arches, in crocodile, for example in reptile, form this quadrat bone of the upper jaw and the articular bone of the lower jaw. So here you see slowly you get 2 structures that are supporting the 2 jaws, and that is kind of their importance for supporting the jaws is going down from here to here. And when you go to human, they are not there as supporting the upper and lower jaw. Instead, the same group of pharyngeal arch cells form our incus and malleus, the middle ear bones. Okay. So this is how a common early embryonic structure has been altered during development to develop species specific structures. Okay, that is how adaptations come. So this is one of the major outcome of comparative embryology, and you can also say evolutionary embryology. Okay, so here you see that these 2 fields sort of we are merging, we are not seeing as 2 separate headings. So and we see one more example. So before we get into this example, couple of important definitions to remember. They are homology and analogy. Okay. So when we say homology, like for example if you take human arm and sea limb or bird wing or bat wing, these are all 4 limbs, okay, modified into hand or this this fin like structure for swimming, or bird like bird's wing for flying or bat's wing for flying. Okay. So common ancestral structure you call homology. So by that definition, our hand and bat wing and bird wing are homologous organs. But if you look at bird wing and bat wing, they are analogous. So they are doing same function. Okay, they both are wings, but one wing did not evolve from another wing. Okay, bat wing did not come from bird wing, or vice versa. Okay. So as wing, these 2 are analogous organs, but as 4 limbs they are homologous organs. Is this clear? Okay. So this is one of the best examples to explain homology and analogy in developmental systems. And the second point we are going to talk about is continuation from the previous one, that is alterations in development helps in evolution. Okay. So here if you look at it, so our fingers, so you have 5 digits, yeah. Convergent means completely two different evolutionary origin, but given the environmental context where they have to fit in, independently they have evolved the same structure. So that is what is convergent evolution. So if you look at the 5 digits, okay, or the 5 fingers, the same bones here you can see, it's just that they have divided and grown more rapidly, so they are longer, and the wedges that existed between the digits disappeared here but did not disappear here. So only those 2 alterations were required to make a bat wing. Okay, the fingers had to rapidly expand, grow with more divisions, and even there is an asymmetry, like one of them is really long and the other one is short, but this web did not disappear. While in our case it was originally there, the webbing, and that helped in the asymmetry of the 5 fingers, but for the fingers to function independent of each other, the web had to disappear by cell death. Okay. So we will later learn programmed cell death. This is an example of, so essentially you are sculpting a structure. Okay, you are removing things from what already exists to create a structure. So that is the thing. So developmental alteration enabling different adaptations. Okay. So here is another example. It's pretty much the same theme, but instead of human we are looking at the embryonic development of another mammal, where we are looking at mouse. Okay. So the panel A shows you in one of them up close, in an fully developed one, the 5 digits, and the corresponding ones are here. Okay. So the smaller, the little finger is the one here, goes very long. And if you look at the embryonic development in the lower panel, you see that very initially they both are like bud coming out of the embryo, and as you go, like particularly you pay attention to this where there is a white arrow. So in the bat you have the web and it is disappearing in mouse, but if you go to the previous one, the web exists in both. So in one it had to disappear so that the fingers can move independently and you can grab things more readily, while this one wants to have the web to use it as a wing to fly. So alterations in development helps in evolutionary adaptation. So the next point is slightly variation in the theme. Essentially we are stating the same in a different way. Here the developmental variations or variations during development or rather changes in development provide the required variations for natural selection. Okay. So here it is not natural selection, it is artificial selection. So actually you need to imagine the socio-political context of the world in which Darwin published his book Origin of Species. So he did not want to immediately in the first sentence say that one species change, they are not fixed. He did not want to say that. So he built a long case about the possibility that developmental structures, organism structures do change, and to make that point he gave lot of examples of artificial selection. How by breeding, selective breeding, we made quite a few plant varieties for our crops. Okay. And similarly animals, and particularly he goes in great length about dogs. So here we are looking at a dog which has really short legs. That is because the hunters who wanted to hunt a small animal called badger that goes into narrow tunnels. So they wanted a dog that could go through the tunnel and catch those badgers, and for that they preferred the ones having shorter leg. So they selectively bred every time looking for the one in the offspring having a shorter leg than the parents. So that is where there is a direction there. Okay. And then they eventually find one that is shorter. So now as developmental biologists we look at how could that happen. So all you needed is an extra copy or overexpression of a certain factor that stimulates differentiation. So when you have too much of that, before you make enough of those cells you differentiate. So, therefore, you make a shorter organ. Had you made more cells and then differentiated, you would have had a longer leg. So, one gene, okay, so change could provide that variation that was required for the selection. And you look at the other one, again a single gene was enough. This is not to mention that single gene changes are always enough for changes, okay. These are because most of the time we believe how can this bigger change happen, right. So, that is why to highlight that point examples where single gene mutations people take. So, here if you look at another gene FGF5, its alterations in its expression leads to follicle development where it produces lot of long hairs and you get this, okay. Selectable variations through mutation of genes that work during development. So, it is kind of already telling you that genes control development, that is a theme we will get to later. And it is not that it works only in dogs, and so look at here. So, this is an FGF5 mutation in patients leading to really long eyelashes and lot of hair in the forehead and cheek, you see this. So, this again is coming from the same mutation, okay. So, this we are going to touch upon a little later as animal models for studying developmental abnormalities in human. So, for example, if you were to see this first in human and you want to understand the basics, and if you are able to make this dog, then you can work out the mechanism here more readily than in dogs, than in human patients. So, the main point is developmental changes or alterations of development can generate the variations on which natural selection can work. So, that is the summary of this the main point. So, these sort of variations observing in animals is okay, but is there any hope for humans? Can we see developmental abnormalities in humans? We do see, there are birth defects and there is something called teratology, okay. So, we are going to look at the teratology now. All right. So, here is an example, okay. So, a mutation in a gene called KIT, which is a transcription factor, causes these defects. The visible defect that you are seeing here is the lack of pigmentation in the forehead and on the abdomen, on the belly of this kid, and this KIT is expressed in neural crust cells, from which, as I told you earlier, the melanocytes, the pigment producing cells come, and also it is expressed in cells that form the ear cells and gut neurons, and as a result this kid has problem hearing as well as malformation of its intestine. And this is also expressed in precursors of blood cells, so blood cells do not form, and it is also expressed in primordial germ cells, and as a result germ cells do not form, and this kid is sterile, okay. This kid is not going to make it up to adult stage, but if it were to, then it will be sterile. And you can have similar defect in an animal where you mutate KIT, you develop similar defects. So, now this becomes an animal model to study a human disease, because you are able to recapitulate the disease symptoms of a human disease in an animal. So, the main point here is the underlying mechanism is probably the same for the normal development, okay. Normally what KIT does for development you are unlikely to be able to study in human, but if the defects are the same in an animal, then you will be able to do it in that animal, and the mechanisms you are going to unravel there is going to apply to human as well. So, that is what we call as an animal models. So, animal models help us investigate underlying molecular mechanisms, okay. So, this piebaldism is a good example of that. Then so that is birth defect. So, this is a good example of actually this is the one for which the teratology title should refer to, okay. So, teratology is usually something that happens to something toxic to embryonic development, okay. So, like for example, poison that kills us or causes problems. but there are drugs or environmental conditions that can affect embryonic development. A classical example is what happened in the early 1960s, where pregnant women were prescribed a drug called thalidomide. It's a mild sort of painkiller to avoid morning sickness. So, here the point is that this actually changed, this one episode changed the way we test new drugs for safety. So, before that people look at whether there is any problem with adult or animals, okay. So, and when there is no problem, then you say that is safe, but then there are drugs that can affect embryonic development. So, thalidomide is a good example. So, the problem with the thalidomide is this: it affects the long bone development of limbs, and that is why you see this man's hands and legs, they are extremely short. And by talking to the the women whom this was prescribed, when they took it, how long they took it, and matching with their the gestation period, they came up with this graph. So, if you take 34 days after the last menstruation, okay, let us say 34th day or within 34 days of conception, then you have problems with ear development and so on, depending on when she took, the embryo developed these problems. But after that it is actually safe, there is no problems. And now actually thalidomide is coming back because it is useful for treating other things, except that you can't prescribe to pregnant women. So, sample size, I think it is about 400 individuals were born with this. So, this was not an experiment that was done, okay. So, here based on the outcome you go back and look at what actually happened, okay. So, this is what people often do to study many human diseases, and that is why the medical record keeping and family tree are very helpful to find gene disease link. So, this is thalidomide is a teratogen because it affects embryonic development. So, these are helpful to understand development. So, now let us move on to our next topics, like we will go slowly towards morphogenesis. So, now in the embryo the cells are not static, they are just not there. So, they are extremely dynamic, they do a lot of different things, they divide and they divide at different rates at different parts, they migrate, they secrete, they change shape, they stay together as a group or they mix with other groups, all those changes happen. And those are listed in this table which goes into 4 slides. I am not going to read out all of them, but the main points are we have 2 kinds of major kinds of cells. One is mesenchymal, where they are not attached to anything and they are they are non attached, okay, cells, and they readily migrate. And so, within that if you look at it, so you have condensation to make a particular kind of structure, so it is a morphogenic event. Then you have division, they make lot of cells like limb mesenchyme as an example. Cell deaths, cells do die, like interdigital mesenchyme we already saw in human digit development. Then you have more migrations, heart cells migrate, germ cells migrate over a long distance to get to the somatic gonad. Then secretion of matrix and also sometimes degrading the matrix, both are essential for development, okay. One good example is cartilage. So, the cartilage is actually secreted part of this, and ECM, extracellular matrix, that is essentially secreted from cells, and sometimes getting rid of them is important if migration is what is required for those cells in the next stage in development. Then growth, some cells simply without dividing their size enlarge, a good example is our adipose tissue cells, fat cells. Then another kind of cells are the epithelial cells, where they are attached, okay. They may be attached to a basement membrane as well as they are attached to each other, and these are the ones they make sheets and tubes, okay. Here is cross section of a tube, and these cells oftentimes, for example during cancer genesis, these become mesenchymal, and that is how metastasis happen. Okay. So, they call that as EMT, epithelial to mesenchymal transition, and understanding how that happens is an active area of research. Delamination, becomes epithelium is like part of epithelium becomes mesenchyme. Then you have shape changes, they remain attached, but then they make different shapes, like for example the lung formation happens by that way. So, these are the different things that happen. So, the summary is that cells are very dynamic in the embryo. So, if you want to follow what structures come from what cells and what those cells had to undergo in terms of this dynamics, you need to trace them, you need to follow them, and that is called lineage tracing. And when you have traced lineage of let us say a very early embryo having very few cells, then based on what structures formed from those cells, you can go back to the early embryo and map different regions of it. From this region this structure comes and so on, and that is called fate mapping. So, that is our next major topic. Okay. Probably this is the only topic we will do today for the rest of the time. So, I will explain as we go. So, this is looking at very early embryo of 4 different organisms. Okay. So, all vertebrates, we are looking at early stage, and these colors tell you. So, this part is going to form the epidermis of the skin, and then like it is not visible in this, but here this is the neural ectoderm going to form the central nervous system, and this is going to form the notochord, and this is endoderm. Okay. So, that is what is mapped. What it means is cells in this region eventually give rise to endoderm. Okay. This is what is fate mapping. So, you are mapping an early embryo, what kind of structures will later come from that part. It is clear. So, this is what is a fate map, and when you look at it, there are some conserved features that are obvious, like one, I will just point out one. If you look at the notochord, in all of them it is kind of in the center of it. So, we are looking on from the top, okay, we are looking at the back, not the ventral side. Okay. So, it is on the top center is where you have the notochord forming, and that is right adjacent to the neural ectoderm, from which you have the neural tube formation that we saw in the frog life cycle. So, this structure. So, see, this again tells you the early similarity among these vertebrates. So, now we look at how do we do this mapping. Okay, what are the various techniques that allow us to trace the cells. So, we are going to again look at it historically, so that we understand how it all began. So, Conklin took an organism called the sea scot. That is because their early embryo have fairly large cells, and different cells, basically blastomeres, have different colored pigments, and therefore you can observe where those pigments go as cell division proceeds, and therefore he started with those. So, it is direct observation. Okay. So, you can do it in many organisms, direct observation. For example, C. elegans, you can do that. Direct observation means not with the eye, but you need a dissecting microscope to look at and observe them. So, this is the map that he finally arrived at. Very early stage, if you take, so this is the animal pole and vegetal pole, and he was able to map the future ectoderm to the animal pole and endoderm to the vegetal pole, and then the other structures as labeled here. And this is the like 16 cell stage, I think, or 8 cell stage. So, where you have individual cells already having like this is going to make only the epidermal ectoderm, and then nervous system will come from A 4.2, and then endoderm. So, this is not going to make only endoderm. From this cell you are going to get cells that form the notochord as well as part of the nervous system also. So, and muscles come from these and muscles do not come from this. So, if you take it out, nothing happens to muscle development. Okay. So, and then you move on. Okay. This is a complex set of this. So, here this vertical line is each division and the horizontal line are the two, and you usually this is not drawn to scale, usually this horizontal length indicates the time taken for that cell division. Okay. So, this is how they draw a lineage map. So far we have complete lineage for only one organism, okay, one species. Anyone knows that? People got Nobel Prize for that. C. elegans. Drosophila is way too complex, you are not going to do it in the near future, its brain has million cells. So, C. elegans, adult C. elegans hermaphrodite has 959 cells. So, we know exactly these 959 cells from where they come from the zygote to the 959 cells, and that is the process, this lineaging of an organism in an attempt to find the entire lineage of an organism. They thought that once we know the whole set of cells and their origins, it will be easier to tease out its development, and in the process they ended up finding a cell. For example, if I were to use this as C. elegans lineage, let us say AB2 divides into A3 and B3, and then this divides in this and this divides in this and then this divides this. Embryo after embryo you see exactly the same pattern, and in one first embryo when you find when A 6.1 divides, for example A 7.1, and that died. Then the scientists thought, oh, maybe I messed up something while doing it, and then you go and redo it again for n number of times, and every time you are able to clearly predict A 7.1 is going to die, and that is when they realized it is not dying by accident or disease or injury, it is programmed to die. So, that is how the web disappeared here to make the fingers independent. So, that is what we call programmed cell death. So, this is an example of unintended discoveries that happen when you focus on basic science research. Okay. So, that is a point away from the developmental biology here, but it is a good example. So, this is how the initial fate mapping and lineage tracing was done, and this works readily where you have large blastomeres and each blastomere has distinguishable pigments and the cell division is invariant. What I mean by invariant is the pattern of cleavage and position of cells and the number of cells are constant from one individual embryo to another individual embryo during development. Okay. That is obviously not the case in a complex organism like human, right. We are of different sizes, right. So, not necessarily the number of cells are identical or the cleavage is invariant during our embryogenesis. So, this is the final. So, it is the a fully developed C-scout embryo where they have done a dissection and photographed it using confocal microscope, and the different structures labeled here have been distinctly colored, and this could be achieved through what we just saw, through the lineage tracing or the fate mapping. So, that is how we know this structure is the notochord in the embryo, and this structure is what is going to form the neural tube and so on. Alright, so that is okay for tunicate embryo where pigments helped. What about other organisms? So, this is an attempt by Vogt. What he did is he mixed agar with different dyes, and these dyes we call as vital dyes. So, what vital dye means is it is a dye that is taken up by a living cell and does not harm the cell division or the development of the organism. So, essentially you can label certain parts of the living organism by providing this dye. So, they do not they do not kill the organism. So, one common example that we do in many labs is looking for apoptotic cells. So, we incubate cells or feed the organism with a vital dye called acridine orange or cyto12, and only the programmed cell death cells are going to take it up, and then you will be able to see them. Oh, these are the cells that are undergoing apoptosis. So, that is a vital dye. Vital dye labels living cells. Okay, that is the main point. So, he mixed vital dyes with agar and allowed it to dry and made small chips of this agar, which is impregnated with the dye, and gently placed on the embryo. This is a frog embryo, amphibian, in different parts, different colors. And once the cells took up the dye, he removed the agar and watched what happens to the cells or group of cells that he has stained with these dyes. And you see, this is the dorsal lip where the cells are going to go in, and then you see them as the cells multiply and migrate, you follow with the dyes. So, this is a dorsal view of the neural tube, these are the two neural crusts, and then when you take a cross section and look at the other, you cut it and you see the other side, lateral view, and then you see where they have migrated. So, cells in this region migrated in this manner. So, this is another way of lineage tracing. So, one problem with this is as the cells multiply, these dyes get diluted and eventually it becomes difficult to see. So, a modification of this is to use fluorescent dyes, which are lot more intense and therefore last little longer, and that is shown in this zebrafish example. So, here a group of cells in this region, what you see in the left panel, the right panel. So, you have green color, this is a fluorescent molecule that is injected into the cells there, and then you see what happens, and later you see here it is into the midbrain and forebrain area of the neural tube. So, that is where these cells migrated to. And using different color dyes and labeling different parts of the embryo, they are able to map in this manner. So, these regions of the early embryo develop into these structures later on. So, this is fluorescent labeling, which advances a little bit more than vital dyes, correct, but then they did not do a very extensive and sophisticated mapping with this. So, these were like initial observations with embryos like frog. But then quickly fluorescent dyes, and right now you use more sophisticated genetic methods. So, we will see that as you go on. So, here they take advantage of similarity in closely related species. So, here you take two boats, one is quail and another one is chick. So, at a more or less similar embryonic stage, you take quail cells from this region and put it on the similar region in the chick embryo and watch what happens. So, here you are able to follow primarily because of two things illustrated here. One is you have an antibody that recognizes only the quail cells and not chicken cells, and you see that. So, this is the neural tube here. So, the neural crust has fused here, the fold has fused, and you see quail cells put there, actually they are here. And another thing is looking at its cellular morphological differences. The quail cells have little larger nucleolus, and that helps in identifying quail cells from chicken cells. And you can extend further, like you can use radioactive labeling, and here one embryo is grown in radioactive amino acid containing medium, and the other one is without radioactivity, and then you take and transplant similar structures at the similar stage, and then you see where it migrates, and that is how they found the melanocytes, okay, this epidermis that cells in the epidermis that make the pigment, they actually come from neural crust. And then you have other cells like somatic gonadal cells, glial cells, these come from neural crust, and this also shows cells migrate extensively. So, a little bit more advanced is here. So, here actually what has been done is one chick which has pigmentation all through, and its part has been taken and transplanted into the other one which did not have any pigmentation, and now you wait and watch when it comes out, and you see this part of the body where you have the pigmented cells. So, you can get all the way up to this stage where you started and what cells contribute to this. So, a last example of this is the genetic way of doing it. So, on really difficult situations, this is what really helps, and this is one difficult situation. Like for example, tadpole, if you remember metamorphosis that we learnt, it does not have any bony structures, instead it has flexible cartilage. But then the frog has bone, it has jaws and it has a head, right, the skull. So what is the embryonic origin of the skull cells? And for that what they did is they expressed a transgene using a promoter that expresses in all cells and made a transgenic frog. Okay, this is a tadpole stage where it is expressed in all cells. Then you take those cells and transplant into another frog. So here you are not looking at closely related species or anything, it is the same species, you can even have isogenic situation, and you transplant it and watch through the tadpole and the adult frog, and then you look at in the chimera. The chimera meaning you have taken cells from one and put in the other one, and then you see this bone developing here. This cartilage as well as this lower jaw actually came from cells that they took from the neural crust area. So even very hard to trace cell lineages can be done using these genetic methods. Okay. So here this gene, this is a fluorescent protein that is not an endogenous, meaning not part of its genome, it is from another genome and you are introducing it here, and such manipulations or such genes we call as transgenes from another organism. But it is worth the effort, then finally you know what comes from where, and then you will be able to trace and then understand the mechanisms involved in development. Cut paste is basically surgery. So you need to open up the embryo and then you cut and do it. So this is one thing I have not done at all, because I worked with invertebrates where you do genetic modifications without really doing surgery, but people work with frog, they definitely do this. So I think we will stop here today with the fate mapping. So in the next class we will continue to enter into how development is controlled.